All right, welcome to the August 2021 MycoTalks. This is Jay Coles. I'm proud to co-chair this session with Professor Neil Gao. We have two excellent speakers for this month's MycoTalks. The first is Dr. Bridget Barker from North Arizona University, investigating the genomic landscape of Coccidioides, and then Professor Frank Van Deverne at Radboud and Nijmegen, and talking about immunotherapy for fungal infections. This is a series that's with the organizing committee highlighted here to provide monthly state-of-the-art talks in mycology. The philosophy is fosters of this session, these series of talks fosters diversity, including career stage, gender, geography, and field. We have two 30-minute talks on all aspects of medical mycology for the research community, followed by 30 minutes of in-depth discussion. We will be submitting, you can submit questions through the Q&A function, which should be at the bottom of your Zoom screen. And we'll collate those and provide those to the speakers so they can ask them at the end of the in the Q&A session. Recordings will also be available on the MRC Center for Medical Mycology YouTube channel. So next 2 months MycoTalks, just a reminder, you do have to register for each session. On September 30th, in 2021, we have Karl Kuchler from the Medical University of Vienna in Austria, and then Dr. Kirsten Nielsen from University of Minnesota in the US. And then on October 28th, we have Betty Wu Shea from National Taiwan University College of Medicine, and then Salome Lubegant-Landemann from University of Zurich in Switzerland. We also have, there's a medical mycology trainee seminar series for trainees. This is calling for all students, postdocs, and clinical trainees, as well as new faculty. That gives you an opportunity to present your work virtually into the global mycology community. These talks are on the second Thursday of the month, but timing can be adjusted to fit speaker and time zone, given the global nature of this series. And it also helps us highlight the scientifically broad and international medical mycology community by nominating speakers with wide scientific interest in global diversity. And the link is here for those that are interested. And I think we'll also put that in the chat. So next, I'd like to turn it over to Dr. Adil Awars, who will talk about this upcoming session here.
Thanks, Jay. And indeed, it's a great pleasure to announce a new seminar series, which we will be running, and which will be launched on Friday, the 3rd of September at 4 o'clock British summertime. And as you can see, we have a great organizing committee together with fantastic colleagues from all over the world. And at each mycoclinic, two clinical experts in medical mycology will be presenting a clinical case study with diagnostic considerations, treatment deliberations, follow-up and outcomes, concluding with a discussion and overview of the relevant literature. And it's really a great honor for this first session to have two great speakers, Chu Lee from Duke University in the United States and Ilan Swartz from Edmonton University in Canada, and they will be addressing recognition and management of fungal diseases in HIV patients. So please have a look at our website and sign up if you're interested to join us for this new MycoClinic series. Back to you, Jay.
Great, thank you, Adil. So with that, I'll turn the session over to my co-chair, Professor Neil Gao, who will introduce our first speaker today.
Well, welcome to you all from wherever you're joining us for the first of the two MycoTalks. And welcome and thanks to Bridget Barker, who's going to talk to us today. Now, many of you will know a bit about Bridget's work. She trained originally in University of Arizona in Tom Mitchell's lab, then moved to Montana State University, where she was working with Rob Kramer, did some work in Aspergillus then, and then came back to Arizona, to Northern Arizona University, where she's been an assistant professor and recently promoted to full tenure associate professor last year. And her work really has focused on valley fever, and she's really known for her work on Coccidioides species. She's worked in a whole series of areas in these organisms, disease dynamics, variation between populations and species of Cocci. And recently she's expanded her interest into the microbiome in human health and has started to turn her attention also to Histoplasma capsulatum and some other organisms. And she is noted also for the novel discovery of the sexual cycle of Coccidioides. And today's talk, Bridget tells me, will be a combination of genomics and ecology, and her title is Investigating the Genomic and Ecological Landscape of Coccidioides species. And please remember to submit your questions, but note we will have questions at the end of both talks. So over to you, Bridget, and thank you and welcome.
Thank you very much, Neil, for that kind introduction. I'll get started with my slides here. Hold on. There we go. All right, so thank you to the organizers for inviting me to give this presentation. It's really quite an honor, and I know this is probably the first talk on Coccidioides, so I'll spend a little bit of time introducing the organism and some of the grand challenges that we face. And as Neil mentioned, in my lab, we really do focus primarily on the ecology and genomics of Coccidioides immitis and Coccidioides posidossii, which are the two fungal species that cause coccidioidomycosis, which we also shorten to valley fever. I have worked in other aniganalian fungi, so Histoplasma and Paracoccidioides, both mostly focused on whole genome sequencing and population genomics. So just to kind of introduce you to the organisms that I work on and why I find them fascinating. This is a group of organisms that contain multiple families. So the order Aniganales contains multiple families within that order. Most of these organisms have enzymatic pathways breaking down keratin, a complex structural protein. They oftentimes have very tight associations with animal-derived material. So we have genomic support for that. Also, where we find the organisms in the environment, they oftentimes are associated with various animal material. They can colonize both dead and living hosts, and they cause a wide range of disease. And one thing that I really want to point out here is this is a lovely phylogenetic tree with some very important information about the family Yellowmycetaceae, which contains blastomyces, histoplasma, etc. But Coccidioides actually exists far removed from that particular family. It is actually in a completely different family, the Aniganaceae. And these organisms are separated by 40 million years of evolutionary processes. So we can imagine that the evolutionary trajectories of these two families are very distinct and unique. So we can learn some interesting things about Aniganales broadly, but I think for understanding the biology of Coccidioides specifically, we can't really just assume what we learn in, say, Histoplasma is going to apply directly to Coccidioides. This order is sister to the Uroshiales, which contains Aspergillus and many other important animal and plant pathogens. And broadly, this is all under the umbrella of the class Uroshiomyces. So as I mentioned, there are two species, Coccidioides immitis and Coccidioides posidossii. So work that we just recently published shows that then in addition to genomic differences, there are very distinct phenotypic differences. And specifically what we tested in this paper would be the growth rate on solid media. So radial growth rate is very different between the two species. So Posidossii actually grows faster at 37 degrees than immitis, and on average actually grows slower at that lower 28 degrees Celsius temperature. So again, this we don't know the mechanism behind this, but it is a very distinct and significant growth rate difference under different temperature regimes. We also take comparative genomic approaches. So looking at genes that are gained or lost, or gene families that have expanded, so a greater number of those gene families. And we can see this here when we compare. So this is from early work that I was involved with. So Tom Sharpton was the lead on this, so doing comparative genomics between Coccidioides, Aspergillus, Histoplasma, and the Fusaria. And so we can see here in this box that there are no genes within these particular families, and those particular families would be associated with the breakdown of plant material in the environment. And in this gray box are genes that would be more associated with, say, keratin breakdown or other sort of host mammalian sort of specific interactions. So we sort of joke that, you know, Cocci likes its bacon and not so much its salad. But really the knowledge gap here from this, so we just had 2 genomes when we did this particular comparison, and we're sort of lacking what the population level differences might be for gene gain and gene loss patterns. So that is work to be done. And to start doing that, we've done a collection of many isolates of Coccidioides across its geographic range. So this tree represents sort of where we are right now. So we've sequenced about 240 isolates to date. Most of those are clinical, but we do have soil and other isolates from veterinarian samples, from zoos, et cetera. And what we quickly notice is that when we apply phylogenetic analyses to these SNP data, we have a very clear and distinct branch between Immitis and Posidoceae. And then within Posidoceae, we have further biogeographic patterns. So there are actually specific genotypes more associated with the soil and patient isolates from Tucson, Arizona versus Phoenix, Arizona. So this is a little bit surprising that we discovered this. We really thought that Arizona would be kind of a unique homogeneous population, but there is some population subdivision between these 2 cities that are within 100 miles of each other. Also, we have further support for this very distinct branch within Posidoceae, which contains isolates from Texas, Mexico, and South America, as well as this somewhat unique, what we call the Caribbean population. And those are comprised of isolates from Venezuela, Guatemala, and a few from Mexico. So we further support that there are biogeographic patterns in our data, and that's even reflected with clinical isolates as well as soil isolates. Another key point is, you know, we obviously study these organisms because they cause disease in humans. And so we've worked a lot with the mycology branch at CDC to investigate these patterns and try to really understand what's the actual disease burden of Coccidioides in the United States. So these are just data from the United States. So it is a reportable disease, but only about half of the states in the United States report the disease. So here you can see the reported cases sort of peaked in 2011. They're going back up again now. So we're still waiting on data from 2020. So COVID has sort of messed with some of the reporting and ability to analyze some of these data. So we're waiting for those, but we kind of expect that we're going to see another one of these peaks. And these peaks sort of have topped out around 22,000 reported cases. So how can I possibly claim that there are 350,000 new infections? Well, we base this on the fact that 90% of the cases are probably asymptomatic or low symptomatic. And so those patients wouldn't actually get the full diagnosis that they had valley fever. So we think that this 22,000 really only represents about 10% of the overall infections. In addition, Texas probably has as many cases as California and Arizona, but they do not report the disease to the CDC. So we would think that maybe these symptomatic cases would be closer to 30 to 35,000. And that's where we come up with our estimate. So you may have noticed, I mentioned that 2011 was a major peak in disease. And so why do we think that that was maybe a hot year for Coccidioides? It was reported the largest haboob. So I don't know if any of you have ever experienced these giant dust storms that occur in southern Arizona, but it was impressive. And there were thick layers of dust throughout the valley, so piled up on cars, inches thick. And so we don't know necessarily if this is the cause of that peak, but it certainly suggests that climate and environmental changes and increasing monsoon seasons could potentially either influence the growth of the organism in the soil or just increase these dust events and exposure. And so why do we care about this, and why is this important for understanding disease? Well, this is an environmentally acquired infection. So we have infection results from inhaling arthrochonidia, which are in the soil, in the dust, and generally floating in the air. And that's why we care so much about understanding the ecology and environmental reservoir. There's no evidence for transmission from infected hosts, except in extremely rare cases. So these would be transplant from patients that had a previous infection, and they don't necessarily screen tissue for potential Coccidioides. And also the data on the environmental niche is really limited. So this limits our ability to do really good climate modeling, and it limits our ability to predict when we might see more risk for disease in the general environment. This pathogen also infects seemingly healthy mammals. And so I put reptiles as a question mark because that's something that we frequently discuss. We don't know if reptiles can be infected. It seems that it's a low likelihood, but no one's really looked. So we try to keep an open mind when we think about that. It also has a dimorphic life cycle, right? So in the environment, it grows as a saprobe, it grows as a mycelium. That mycelium, every other cell in the mycelium matures into an arthrochonidia, and that arthrochonidia is what is the infectious propagule. So when the dust is disturbed or the soil is disturbed, the host is potentially in contact with those asexual arthrochonidia. In the host, the organism rapidly gets signals from the host, and it changes its morphology into the spheroidal structure. So it goes from this polar growth that would be in the soil to isotropic growth in the host. These large structures round up and internally develop endospores, which can then propagate the life cycle. So each endospore can then become a new spheroid. It's 4 to 5 days from a spheroid initial to rupturing spheroid. These endospores can then disseminate throughout the body. There's also good evidence for latency. So in humans, we see patients that would have been infected 20, 30 years prior have a reemergence of the infection. And we believe that this is due to a latent infection. Immunity, so there's cellular immunity in humans and in mice. And that we have good evidence that there would be potential for vaccine. The early innate immune response, so those early interactions with the arthrochonidia developing into that first spheroid and first rupture, those processes are not as well described, but people are working on that. So I propose that as the medical life cycle, and I want to just propose another potential way of looking at that life cycle. So John Taylor at Berkeley and I have collaborated for many years on the environmental life cycle and the soil ecology of the organism. And we proposed this hypothesis to kind of explain what this organism might be doing in the environment. So we believe that in addition to humans, small mammals in the desert are susceptible to an infection. And this could be either through long distance dispersal in the air or short distance dispersal in the air, but regardless, then this naive animal is exposed to these arthrochonidia, and within that living mammal, the spherules will develop. So there's two potential options here. So either the spherule develops, it causes severe disease, disseminates and kills the animal rather quickly, or potentially there's a latent phase and the organism stays viable within the animal tissues. The animal dies regardless, so that the animal will die eventually. And from that dead mammal, the hyphae can grow and produce that additional round of arthrochonidia. We believe that this is really important to consider because the disease is not transmissible. If the spherule cannot get out, if the fungus cannot get out of that mammal to infect the next host, then that sphereal would be an evolutionary dead end and would therefore not exist. So that's our proposal. And also we have additional support for the host or potentially the host environment. So that burrow environment being the site where we believe the organism is most prevalent. Because burrows themselves and animal derived, so grave sites, archaeological sites are often point sources of infection and outbreaks. And the burrow site itself represents the majority of our positive samples. So I spend a lot of time hiking around the desert, collecting soil, looking for this organism in the environment. And when we do that randomly, we don't find very many positives, but when we target animal burrows, we definitely get more positives from that. And this is a recent publication from a master's student who finished up last year. And if I would encourage you to read that if you're interested in that story. So why do we believe that understanding these processes and doing genome sequencing and understanding gene gain and gene loss, why do we believe that they provide clues about general life history and evolution and the potential for outbreaks? So again, the global epidemiology of Coccidioides, we have a high concentration of disease in California and Arizona, Texas as well, although they don't report. And then Mexico also has a high burden of disease. There are small discrete populations in other areas. So we have in Venezuela, Guatemala, Eastern Brazil, Argentina, we have small discrete populations where we find the organism and we see disease in the population. These overlap with desert areas. So here on this map, I've shown, so in the brownish color are sort of more cold deserts, and in the orange are more hot deserts. So we definitely see Cocci more in the hot regions of these desert areas in North America. But here we also have one discrete population in Eastern Washington. So that would be represented by a cold desert. So we believe that there is potential for this organism to exist in these cold desert environments. And then potentially with climate change, these deserts might begin to be warmer. So does that, does that indicate that there might be more potential for outbreaks in regions that are not considered to be currently endemic? So again, we look at the natural genetic variation of these isolates, and the things that we're looking at are sort of novel genetic variation, so potential gene gain and gene loss within these populations. As I said, we've looked at the species level and comparing to very distantly related fungal organisms, but will we find patterns that help us understand clues about how this organism could potentially spread to these new areas, looking at host specificity and ecological niche by using genomics. And so again, the reason that we are somewhat concerned about this expanding region is a recent publication with Morgan Goris and her advisors, Kathleen Tressiter and Dr. Randerson, Jim Randerson, would suggest that mean annual temperature and mean annual precipitation are key drivers for the distribution of Coccidioides. And so as the mean temperature increases in the United States and precipitation, precipitation patterns change, could we potentially see expanding habitat? So it's predicted that by 2095, that both thresholds for temperature and precipitation that seem to potentially be associated with Cocci presence in the environment, both thresholds will be met throughout the Western United States. So what is important about these regions? What are some characteristics that we could look at in the environment that might be related to the organism and maybe limiting its distribution, right? So that's just looking at temperature and precipitation, which are strong abiotic factors, right? So we know that these two factors definitely play a role in the abiotic environment, but there's a lot about the biotic and then other characteristics of the abiotic environment that we don't currently understand. Regions colonized by Coccidioides, as I said, are characterized by these hot, dry, alkaline soils, but they're also occupied by other endemic species. So there are many animal species that are found only in North and South America. So if we believe that the host plays a role, so the natural host plays a role in its distribution, we're interested in looking at what other animals might be present in these areas. And would the ability of these animals to migrate to these new areas be an important consideration? So various reptile species, again, as I mentioned, we don't really know whether reptiles are a potential host or reptile burrows are a potential reservoir in the environment. Heteromyid rodents are specific to North and South America. This would be pocket mice and kangaroo rats. And then additionally, so this sort of bridges the biotic and the abiotic factors. The desert soil has been characterized by biocrusts. So these biocrusts have been really impacted in the Western United States by grazing and agriculture. So we've lost a lot of these natural biocrusts. And so what implication might that have for the spread of the disease and the aerosolization of these arthrochonidia? So we have a grant to look at that. I won't talk about that today, but we are looking at that. And then pH. So pH is kind of what I'm going to spend a little bit of time talking about. So again, thinking about these big questions, what are some genomic clues that might help us better understand the ecological niche? And we believe that understanding that ecological niche is important because it will help us develop more accurate models to predict the response to climate change. So genome analyses can help us define hypotheses. Oops. Okay, now it's not wanting to go. There we go. So again, thinking about some strategies for choosing these targets, we know we can look at genes that are upregulated in vivo. We can look at genes that are upregulated in the serial. And again, we can look at gene gain and gene loss or gene family expansion. So I worked together with Jason Steich and Marcus Teixeira to really improve genome assemblies. We've done a lot of re-annotation. We're in the process of publishing some of that work. So again, thinking about in vivo, there are some good strategies. We're thinking about in vitro and thinking about what genes might be characterized in other fungi that are important for these ecological adaptations. So I've mentioned Thermo Tolerance as being different between the species. So another aspect that we wanted to look at was pH sensing. So we've got some great genomic information. We've got some excellent tools in our toolbox to sort of develop hypotheses. But to confirm these hypotheses, we really need to, gosh, I don't know what's going on here. Hopefully I didn't have these timed. We have classical genetic tools in our toolbox to look at the function of genes, and various methods have been used in Coccidioides. So agrobacterium tumefaciens mediated transformation has been used. Protoplast with linear integration has been used. But the take home here is that very few genes have been knocked out and none in the other species of Coccidioides nivatus. So we've turned to developing CRISPR. And so why? One of the challenges of doing knockouts in Cocci is that those arthrochonidia that I talked about are just naturally multinucleate. So when we go to create protoplasts, we generally create protoplasts from that arthrochonidia, or we'll create germlings to do the agrobacterium, and all of those are going to have multiple nuclei. We've understood now that Cocci actually tolerates that heterokaryon state, so it will tolerate having a wild type nucleus along with a transformed nucleus, and so that has made it complicated to purify to a homokaryon. We believe now from work that we've been doing over the last 2 years that CRISPR actually is able to target the multiple nuclei in a single transformation, and so fewer passages are needed. So again, we have a long list of things that we're interested in, what I'll talk about just briefly, and this is very early stages, so it will be very brief. We're interested in looking at the Pac-C transcription factor. So in addition to pH response, Pac-C, RIM101 has various functions in other fungi, potentially sporulation, potentially dimorphic switch, regulation of virulence factors, et cetera. So there's a broad range of phenotypes that we could expect from deleting Pac-C in Coccidioides. So this is a master's project. So this student, Karis Miller, worked on developing CRISPR and was interested in Pac-C. And so, right, it's the PAL pathway that senses this ambient pH, a downstream signal, cleaves Pac-C to make it active. And so we targeted sort of the end of this pathway to hopefully get interesting phenotypes. So we targeted Pac-C for deletion. Phylogenetic analysis suggests that Pac-C should have similar functions to its close relatives based on phylogenetic analyses. We have both copies in RS and Silvera. Silvera is a Coccidioides postadocii strain that is commonly used in the laboratory. So we decided to use Silvera as our strain of interest for doing this initial knockout. So again, to investigate what we might expect, is pH important? What is Cocci's tolerance for high or low pH? And it does seem that from these preliminary investigations that Cocci prefers to grow between a pH of 7.5 and 8. We are utilizing a system that's been used very effectively in Aspergillus fumigatus. So this is out of Jared Wendell's lab. So Quasi has been very kind and answered all of our questions when we've been developing this system, but it's in vitro assembled Cas9. So we don't have an endogenous expression of Cas9. We use the protein itself and complex it with these gRNA duplexes. We've designed a repair template. So we have hygromycin as our selection marker. And then we have 50 base pair regions of homology just outside the cut region. So hopefully, again, enhancing our ability to create these double cuts, remove Pac-C, and replace it with hygromycin. And we use the protoplasting strategy from Chung Yu's lab from her protocol published in 2012. So again, just some preliminary results here. So we believe we have several mutants. We have confirmed this. So here's the wild type. We don't get amplification of hygromycin. Here's one of our strains. We do get amplification of hygromycin. So again, in the wild type, here's the Pac-C locus, and that is absent in our knockouts. And interestingly, we had many. So again, we're optimizing these concentrations of how much Cas9, how much repair template, et cetera. And ultimately, in one of our final transformations, we think we've hit the sweet spot. We actually obtained 48 colonies, all of them have a yeast-like growth with sort of aerial hyphae. So here we see wild type Silvera, so these white fluffy colonies that grow robustly on a plate, versus these Pac-C strains. So they had sort of an odd yellowish color, very glabrous and flat. A few aerial hyphae will start to develop in the center of that inoculation plug, but overall much more yeast-like. We look at this under the microscope and we see very large yeast-like structures growing, not spherules per se, but sort of look like spherules. Again, these strains all are growing a hygromycin and contain the hygromycin resistance gene. We've done RNA extractions and loss of the Pac-C transcript. So right now, what we're doing is looking at southerns to confirm that we have a single integration, as well as doing whole genome sequencing to really see. So again, this is the sort of the first time that we've really done CRISPR in Cocci, so we want to make sure there's no off-target effects or other issues with CRISPR. So we're actually doing whole genome sequencing on some of these deletion mutants. So in summary, I hope I've convinced you that I'm really interested in understanding the ecological niche of this organism. It's really important, I think, because most, not just for Cocci, but for many fungal organisms, these are environmentally acquired diseases. And so understanding the ecology of these organisms, I think, is extremely important and can help us with future modeling of, thinking about future outbreaks, thinking about expansion, and thinking about novel strains that are arising. And I gave you an example of that, right? So soil pH in the desert region is 7.5 to 8. And this is hypothesized that this is one of the factors that supports the growth of the organism in the environment. So is this true? So we need to start working on actually functionally characterizing some of these genes that are known to be associated with pH sensing and regulation. So, we really need these methods to delete genes. We need these well-annotated genomes. We need a population level summary of these genetic variants to really understand and compare sequences on a population level as well as between species. And with that, I will thank you for your kind attention. It was wonderful, again, to be invited to give a talk on the MycoTalks. I've really enjoyed the seminar series. And I thank the people on my team that have really led the effort. And I work within a great center at the Pathogen and Microbiome Institute that really supports a lot of the research that we do and supports our very expensive biosafety level 3 laboratory experiments. And with that, I will pass it off to Neil.
Thank you very much, Bridget. As you know, what we're going to do now is we're going to move to our second speaker of the series, and I'm going to pass on to my co-chair, Jay Coles, to introduce our next speaker. And then please do put your questions for both speakers in the Q&A, and we'll return to those after we've heard Frank's talk. So thank you, and over to you, Jay. Great,
thank you, Neil. So it's my pleasure to introduce our second speaker, Dr. Frank Van Deverne at Nijmegen Radboud University. Frank received a master's from Free University in Amsterdam, and followed by a doctorate work at Nijmegen with Professor Kohlberg, Natea, and Van der Meer on the interplay between innate immunity and Th17 responses in Canada infection, and then followed by clinical training in internal medicine and infectious disease, and then a postdoc at Colorado with the Charlton O'Leary's group. And since moving back to the Netherlands, he's made seminal insights into human immunity to fungal infection, and just a few highlights. His group described these STAT1 gain of function mutations associated with chronic mucocutaneous candidiasis. He's had a longstanding interest in the IL-1 family members and signaling and autophagy in the context of Aspergillus infection, including allergic bronchopulmonary aspergillosis. And then most recently, he's also been doing work on the immunology of COVID-19 infection. Today, Frank's going to talk on his research is really the host interaction with the fungus, and so he's going to talk about immunotherapy for fungal infection. So look forward to your talk.
Thanks, Jay, very much for your kind introduction. Indeed, the journey has been driven basically by understanding disease mechanisms and translating this into therapies. And basically, since we study the immunology and try to understand what it is, we came up with more modulatory therapies that are widely used in oncology and in rheumatology, but in infectious diseases, they are still at the beginning, and with COVID that has changed, and I'm very glad that we are getting more and more insights to translate this to the clinics. So I want to pick up on a story that we did before, and with that I want to introduce a disease, a rare disease that we're studying also for quite some time, chronic granulomatous disease, which is basically, in there are clues of why you become so susceptible to fungal infections, specifically Aspergillus. It's a very big problem in this rare disease, and is due to a mutation in one of the components of the NADPH oxidase complex, and that's needed to produce reactive oxygen species. And if you think about what it does, it makes a lot of sense to think that people with a mutation there, that it doesn't work, would be very susceptible to infectious diseases, because once a neutrophil phagocytoses a pathogen like a Staph aureus or an Aspergillus, a fungus, in the phagosome, this complex will click there and will start producing reactive oxygen species, which can harm and kill the bug inside. So it makes a lot of sense if that doesn't work, that you become susceptible. But along the way, we learned that many more things are disturbed or dysregulated in this disease. One of the things is that they are also very inflammatory. They can present with inflammatory problems, and clues to that, we discovered that there are more mechanisms disrupted in this disease. So one of the things that Jay already mentioned was that we found in CGD one of the mechanisms how to clear pathogens or other things inside of a cell being defective in CGD. So if you phagocytose a particle that eventually will fuse with the lysosome and the content is then degraded. On the other hand, you can clean up your cytoplasm of old organelles or other things in the cytoplasm that should not be there with a mechanism called autophagy, self-eating and surrounding this with a membrane, and then it fuses with the lysosome. And in between, there is a fusion of these two mechanisms called LC3-associated phagocytose, which is basically a boosted phagocytic pathway where components of the autophagy are used to make it more efficient to clear the pathogen. And we found several years ago that that pathway is deficient in CGD. Very strikingly, that LC3 that is needed to form that membrane and also the diffusion of the lysosome is not recruited to the phagosome of the CGD patients. And we found that if you then use a drug called anakinra, which blocks a cytokine I1 pathway, that we could restore. If you look at panel B, the black bars is the same CGD cell, but then in the presence of this drug anakinra, and we could restore that LC3 recruitment of the cell. So that was very interesting as a mechanism. How can such a drug restore such a crucial mechanism? And also, is it important, for example, to clean up your fungal pathways? Here is a experiment where you can see in the black bars, those are macrophages, muromacrophages fed with Aspergillus conidia, and then we look at the outgrowth. Black bar is wild type, but if you look at the NOX2 knockouts and also the ATG7, they basically are not controlling the fungal outgrowth of the Aspergillus, and NOX2 knockouts are the NADPH oxidase deficient cells, so the CGD cells. And interestingly, if you then titrate in anakinra, we could restore also the fungal outgrowth, but that was not happening when a cell was autophagy deficient. So you cannot bypass the autophagy machinery if it's deficient with anakinra, but you can do something in this dysregulated balance of reactive oxygen species that are not produced. So that makes us think, how does that work? How can anakinra do this? And we made the next step to look, and we took those murine macrophages and we did a transcription profiling in the presence or absence of anakinra and see what is really different. And one of the things that popped out, many other things also, but one of the things that caught attention was PPAR. And we know in CGD, and that's been published in 2015 by the group of Stephen Holland, that if you use a PPAR gamma agonist, a PPAR agonist, that you can partially restore some of the phagocytic function via mitochondrial pathways in CGD. So that was an interesting hit to see that anakinra basically could do something there in the PPAR and also something with mitochondria. So we started looking into the role of the mitochondria, and we could see by electron microscopy that if you take a raw cell macrophage and you treat them with or without anakinra, and a few hours later you look inside and you look with EM, you could see that the mitochondria are different. Non-treated, we could see also swollen or enlarged mitochondria, I must say, that are also in some vacuoles of those cells, while this was much cleaner and more viable mitochondria when there was anakinra. So it looked like anakinra did something on the mitochondria and preserved somehow some of the functions that could be different in the cell. Now, one of the status of that redox in CGD is determined by antioxidants, and antioxidants enzymes such as for the mitochondria is very important, is SOD2, also next to SOD1 and SOD3. We also checked those, but we could clearly see that only anakinra had an effect on SOD2. And that was also transiently in a few hours, and then it was the protein was lost again. So it looked like anakinra could do something on SOD2. On the mitochondria, it would make a lot of sense. So we thought, is SOD2 important in the disease CGD? Could we find a hint? Does it have to do anything with LC3-associated phagocytosis or fungal killing? So we moved on and we did an infection in a mouse model. We found that in the naive, there is no infection, and these are slides of the lung histology, and none is where there are, when they are infected with Aspergillus and the wild type mice. And then in green, you can see the SOD2 staining. And with anakinra, we could induce that. And interestingly, also we could induce this in the CGD cells. It might be a little bit less in the basic part, and when you count for that, it looks also less. But the most important thing is that you can induce SOD2 in the setting of a deficient NADPH oxidase. And that made us think that is SOD2 in that sense important. And when we did targeted experiments with siRNA, we could see that the fungal burden was higher if you block that SOD2 function. And we could also see that in the lungs, the LC3 expression was lower when you had this siRNA against SOD2. So these were the first hints, and we are still exploring the specific pathways, how this links up with mitochondria and with the redox reaction. But it looks like anakinra has an effect via SOD2, and that can restore some or partial effects that are important also in CGD. So this is an ongoing story, and I think it's interesting because it touches more upon how that works. And we also observed that many of these things are IL-1 receptor type 1 independent, so meaning that maybe anakinra can do something more than just block the receptor and intracellularly. And these are data that are in development. So importantly, we know LC3 recruitment is defective, and anakinra can restore the fungal killing partially, but also in the setting probably via SOD2, and also protects mitochondria, which would make sense in CGD to have a beneficial outcome. Now, that was one part that we're working on, but we also asked ourselves the question, are there other mechanisms involved? And Marjolina Bruno, PhD student, working on this for the last 1.5 years. And she used several techniques in collaboration with other people to see, are those CGD cells somehow really different also on other aspects? And one of the first tools that we used was single cell RNA sequencing. Here you see a healthy cell population drawn from blood, and you can do by RNA-seq mapping of subsets of these cells and get really nice insights into all those different subsets of the blood compartment. And one question that we asked ourselves, because we always focus on monocytes and neutrophils as an innate immunity lab, maybe we are missing something, maybe we see other cell populations that are very different in CGD. But on the one hand, not unexpected, but also very reassuring is that when you then look in differences between control cells, versus the CGD cells, the real big differences were in the monocytes and neutrophils on the transcription level. On the RNA-seq, it was really clear, and this really made us strongly believe that the two compartments, monocytes and neutrophils, are the cells to look at. So knowing this, we went back and we studied them functionally, and one of the things that we had already observed, but we wanted to test again, is those cells are very hyperinflammatory. So if we give a stimulus, they really make initially more cytokines compared to healthy control cells. And this is true for neutrophils on the left for IL-8 if you stimulate them with PMA, but also for IL-1 beta production by monocytes if you have several different stimuli. Now, having said that, means that the cell is already very primed or very in a status where it can produce a lot of cytokines. And with this, knowing also in our lab that we are very interested in the status of such an immune cell like a monocyte or a neutrophil, this is the concept that Mihai Netea also put forward. We knew already for a long time that monocytes, for example, can have an effect where they have seen LPS and then they become very tolerant. They don't produce cytokines in sepsis, for example. And this is also at the level at epigenetic regulation of the cells, but also the other way around, called a trained cell, where there is a monocyte that when it sees a stimulus, it produces more. And how does that work? It's what's seen by the cell and then imprinted epigenetically what the cell does when it sees a secondary stimulus. So we thought maybe the cells are already in the blue setting because they look like a primed cell, they react more. So maybe they are epigenetically different. And we wanted to do a training experiment with the CGD cells. So how do we do that? We classically have this concept of you see a stimulus that shapes the status epigenetically of the cell. You stimulate for 24 hours, then you wash away, you wait for a week, and after that week, you give a second stimulus and see how the cell behaves. And then you do the analysis. So all the bars that you see in the cytokines would mean that cells have seen a stimulus or not before, and then at day 7, this is where the results are when you come back with your stimulus. And when you do this in a healthy cell, a monocyte, for example, and you stimulate them with LPS on that day 7, where they have seen something like BCG here, or not BCG, which is the RPMI, the medium, you can see that they are boosted in TNF and in IL-6. And interestingly, a CGD cell doesn't do that. It wasn't clear that they produce more cytokines. The trainability basically of that cell was not happening. So we're deficient in the functional training experiments that we were doing. So that made us think maybe they have an epigenetic different profile. And by doing that, we looked at their epigenetic profile. We indeed found that, that their epigenetics is really different. It's dysregulated, it's different in the setting that unstimulated, these are the cells that you just take out of the patients unstimulated, they already have many promoters open to produce or start producing, for example, for chemokines and cytokines. So they are sort of open and in that setting, trained in the setting of hyperinflammatory status. So knowing this and knowing that epigenetics is a lot of regulated at the level of metabolic spectrum, and CGD being NADPH oxidase being also at the heart of several metabolic pathways, we thought maybe the metabolome of the CGD is also different, and that shapes that epigenetic landscape, which is different. Here you can see that, for example, several amino acids or metabolites are very important for epigenetic enzymes that shape your landscape of your epigenetic status. So this in mind, we started looking into more what is the metabolic status of the cells, the innate cells, and we focused on monocytes and neutrophils again. And monocytes, if you measure intracellular the metabolome, you could see basically that the amino acids were lower. So we had clues that that part was really different, and some parts of the glycolysis pathway were also lower. So there is clearly a difference in that status of the metabolome of a monocyte compared to a healthy cell. With the neutrophils, we took a different approach, a bit difficult to get a metabolome out, but we found by using the RNA-seq that I showed you in the beginning and a prediction model called COMPASS to see and predict what is metabolically different in this setting. They could predict that again, the amino acids were really different in neutrophils versus healthy neutrophils. So this gave us at least a clue that epigenetically and metabolically there is a different status of CGD cells. But what about functionally? You can test functional capacity. One hand, you can do the glycolysis assays, and then you measure ECAR, which gives you a curve if you put in several components, and you can do mitochondrial respiration. So these seahorse analysis that we did in CGD cell are more functionally looking at the capacity of glycolysis and mitochondrial respiration. And these are very, very, very basic and preserved mechanisms that should be highly preserved in a cell. So we were really struck to see that, for example, in neutrophils, you see the glycolysis with a glycostress test, and we did the ECAR that already basically they are different, basally, I mean, different. And then in those stress tests with the several components, they remain really different than a normal cell. And this is so fundamentally metabolically important that such curves are very striking. And so we saw real functional defects in the glycolysis of neutrophils. In contrast, we didn't see that with the oxygen consumption rate. So if we started looking at the mitochondrial respiration in those neutrophils, it was not as different. More subtle differences that could also be because the neutrophils rely less on their mitochondria for their metabolic status. But anyway, it was a striking observation that it was mainly glycolysis in neutrophils and not the mitochondrial respiration. Knowing all the amino acids that were really different also in the neutrophils. For monocytes, the story was a bit different. They were also for the ECAR was very deficient compared to healthy monocytes, but also the mitochondrial respiration rate was really strikingly different. So these are very broad metabolic defects, functionally also defective in CGD cells. So at this point, we thought, is this then due to all the inflammatory properties that are with the patient? They have several disease status, or is this really intrinsically due to the NADPH oxidase itself? So we used a blocker for the P47, and we could mimic really the defects that we were seeing for ECAR and OCR. So the glycolysis pathway and the mitochondrial respiration were both impaired when you blocked P47 in monocytes and you stimulate them, for example, for Fusaria. You didn't see the things that we would normally see in a healthy cell. So metabolically, NADPH oxidase has a much broader effect, and on glycolysis and mitochondrial respiration, we do see strong effects. Now, one thing that puzzled me already for a while was the gamma interferon prophylaxis that we use also in patients, which really are there not to treat the infection, but prevent the infection. And these are studies from the 90s where they really saw an effect of prophylactic regimen of gamma interferon treatment. And obviously it had effects for preventing infection, but that was not via the induction of reactive oxygen species, because that was defective. So it remained also a question of how does the gamma interferon work. So we thought maybe the gamma interferon also has consequences on these kind of metabolic status of the cells. So what we did is we tested the first blocker for the P47, and we could see that, again, the blocker reduced the ECAR, so the glycolysis, and when you stimulate them with zymosan, then in a healthy cell, it's much higher. Gamma interferon treatment, which are the full dots, could partially restore some of the CGD cells, which are in red, and this means that maybe to near normal levels, but in a control, it goes much higher. So we were really interested to see how does that work in a CGD cell. If you then give gamma interferon treatment, can we restore some of the metabolic effects? Because this hinted that there will be some restoration. So very nicely, when we go back to the experiments, we could again repeat that the CGD cells, monocytes in glycolysis were deficient, but then with the gamma interferon, we could restore it to the normal levels of the curves. So we saw normal curves compared to healthy controls in several parts of that glycolysis pathway that was defective functionally. And the same was true in the monocytes for the OCR, which was even also very striking to see that some parts of that we could restore. The basal effect of the OCR we could restore with gamma interferon pre-treatment, And then also parts of that curve that were really functionally different, restored by gamma interferon. So this told us maybe the gamma interferon has a effect on the metabolic status in a good way, getting more to a normal profile. So maybe that also changes epigenetic landscape. Eventually, something like the training experiments would be affected. And indeed, when we did the training experiments with the gamma interferon pre-treatment, we could see that the red bars, which are now from the CGD compared to the medium, were now induced, and we could see effects. So the training experiments worked in CGD cells with gamma interferon in a normal way that we would expect, giving more cytokines to a restimulation. So we're very eager to know if in vivo treatment, because we treat patients now with the gamma interferon prophylaxis, if they are also in that setting epigenetically reprogrammed in their cells. At least what this learned was that it's a much more broader metabolic defect, and it might give another clue how gamma interferon works in CGD, besides just increasing the fungal killing. Maybe this also is at the heart of the increase of the fungal killing that might be there, but at least the metabolic status of the cells could be changed, shifted, and gamma interferon is a drug that seems to bring that back to normal in CGD cells. Now, having said that, we are looking for next steps. How does gamma interferon do this? We already have some nice clues. We want to study that obviously in the prophylactic, and sometimes we start the gamma interferon so we can compare. But also, can we do this, for example, for other diseases like influenza or COVID-19? Can we take this data that we are learning from the metabolic dysregulation to see how we can immunotherapy modulate those disease status of cells, which would hint to the fact that if you have a metabolic disrupted functional cell, gamma interferon would be capable to increase that. Now, for influenza, we already have a case to look closer because we know that it can suppress the NADPH oxidase complex that we also could mimic in human cells when we looked at ROS production in the presence of the live attenuated flu. We also see lower ROS. So there are several hints to see that a monocyte infected with influenza can have that CGD phenotype. So this is one way in influenza to look. And it's interesting to see if the metabolic status in those cells are also very different. Having said that, we know that the influenza itself also gives the increased susceptibility to aspergillosis, something that we learned a few years ago, but it's becoming more and more consistent over the years that severe aspergillosis, at least here in the parts of Europe where we see it, is about 1 in 5 patients admitted to the ICU. And one of the things that I want to highlight is an entity called Aspergillus tracheal bronchitis, very severe form of that invasive pulmonary aspergillosis, high fungal burden in the airways, impressive histology. And a French group has looked at how often this is the case, and they looked at 213 ICU patients admitted with influenza in that cohort. A small group had already factors that predisposed to aspergillosis, but many were non-immunocompromised. And the total group came here at the bottom, around 21% of those, 1 in 5 again, of the patients having influenza. And then they looked further, like how many patients could eventually be diagnosed by doing biopsies and looking at the trachea for plaques. And out of those 45, they could evaluate 35 for tracheal bronchitis, and 10 of them had that with tracheal bronchial lesions. And if you see them once or more often, you immediately recognize them. And in biopsies, they are very clearly seen as the fungus grows there into the mucosa. But look at the mortality, it's really, really different. 90% mortality in those patients compared to 44%, so about 50% that we already know. So it's an entity that is very important to notice and comes with a high mortality. So for that, we also had just before the COVID a study where we started prophylaxis when there was still influenza. And we did a study which basically learned us a lot, because the study was designed of when you enter the ICU, you have influenza, you get randomized between prophylaxis with posaconazole or not. But we learned that within 48 hours, 16 out of the 89, so roughly the amount of patients that would get invasive aspergillosis already had it when they entered. So we weren't looking at a prophylactic study. We should immediately look at treatment. So it's a very fast infection, and that really made us think, like, if you enter the ICU and you already have that, that's a new insight, also demands a different strategy than just doing this. So a lot to be learned and needed to get forward with the influenza aspergillosis. And then obviously when the COVID happened, we were very interested to see what is that similar, is this happening? And initially there came a study out of Italy where they very nicely and systematically looked at all the BALs in the patients that were admitted to the ICU with COVID. And they came to a huge number, 30 out of 108 having the diagnosis. Yes, it was dependent on the classification, but still was a number like 1 in 3, which was very high. But soon after that, we learned that it depends on the how you define the case, and also in the different countries, there were very many different percentages reported. So in the end, it's somewhere between the 15% to 20% that we are seeing overall CAPA, so COVID-associated pulmonary aspergillosis. But why I'm mentioning this is because I think it's an important part that the viral pneumonitis itself is also, together with the epithelial damage and other mechanisms, predisposing to the aspergillus. And now we are in the stage where we are getting more and more into understanding why that aspergillus is there. And one key thing to come back to, and also why I mentioned this immunotherapy and fungal infection, is that the two components, like the hyperinflammatory part and the immunoparalysis part, where it doesn't function, it doesn't do what it does, or it's working too hard, are both things that are amendable by immunotherapy. Hyperinflammatory, anti-IL-6, anti-IL-1, etc. But for the metabolic dysfunction, as we've seen, maybe the gamma interferon and the boosting of the capacity to kill the fungus. So these two players, and they can also coexist. So we need to find profiling to see what happened. That's what we tried also in COVID. And interestingly, I want to point out that although we do see a monocyte HLA-DR expression, which is lower, meaning that monocytes are less activated in that setting, they are not, like in sepsis, losing their capacity to produce innate cytokines. They're not immunoparalyzed. They make those cytokines. But in the setting of gamma interferon, they were very deficient, the more severe COVID you had. Meaning that eventually the part of COVID playing a role in aspergillosis is on several levels. And one of the things that we looked at is the tracheal bronchitis. This is one from COVID, where we have histology. And the tracheal bronchitis that we saw with the plaques were not as invasive as with the influenza. Here I want to show you that it goes into the mucosa, but it doesn't go very deep. If you look at the influenza, that was just, in all cases that we have seen in biopsies, very dramatically invasive, and also invasive into the vasculature, meaning that there is something different in influenza versus COVID with this severity. Now, having said that, we came up with a model that would say, what is then the background? What is the pathophysiology between this? This is the last part that I want to mention, is the difference between the viral pneumonitis of COVID and influenza. Now, first of all, the genetic polymorphisms that we all carry, or the factors that we are, is not something that would be different. But the infection by the virus itself is different, because the lytic infection of influenza is much more severe compared to COVID. If you do that in cell lines, or in look at the devastation and lytic infection of the virus, it's much more clear that influenza is more lytic. And the cytokine storm syndromes, they could basically be the same. And the lymphopenia could be the same. But for example, in Aspergillus or in influenza, there could be an NADPH oxidase complex suppression. It infects monocytes and macrophages. It's immunologically different. There could be a role for the neuraminidase inhibitor, which I didn't talk about, but it could be several layers. Now, I think the key question is, if we now start immunotherapy, which we're doing for COVID, like dexamethasone, and everybody wants to do anti-IL-6 also now in influenza, what will happen? Where will we stand with the Aspergillus infections then? So we have to learn this mechanism, how this works, and we have to know how to intervene, where to look at it. And in red, we placed where we think the immunomodulatory parts are crucial. And one of the things that was coming more strongly out of the CGD work that I presented earlier is the gamma interferon regulation of the metabolic status of the cells, which could be a very key thing to do. We were always scared that it would drive people into hemophagocytic syndrome or into a macrophage activation syndrome, but we have done it before the gamma interferon treatment for the SARS-CoV. We didn't see that, but we also saw clear good effects on the cells and on the status of the host cells. So it could be an option even in COVID or in Kappa to do a study with gamma interferon. And I think that's where we want to go. Also, we're setting up immunotherapy trials for that, but specifically also on viral pneumonitis with the Aspergillus. The dexamethasone, the gamma interferon, anti-IL-6, anti-IL-1 are really key questions at this point where we are trying to find answers. So with that, I would like to thank many people that contributed to all these stories, especially Marilena Bruno, who did all the work on the CGD cells and the metabolism, and Intan Dewi, who did a lot of work on the influenza-associated aspergillosis, and all other people. Thank you very much.
Great, thanks, Frank. So we'll now move to the Q&A session. Professor Gao, do you want to kick us off with questions for Bridget?
Okay, and please do continue to drop your questions into the Q&A box. And what we will do with Jay and I is we'll try and organize those so that we'll try and get through as many as we can. We may splice some together, which might mean that your question isn't in the order in which it was sent, so don't get worried about that. And we'll take questions alternating between the speakers. So we start, Bridget, for a question for you, and it's about co-infections with viruses in the host, in the rodents. And the question from Ted White is, can you say anything about co-infections in rodents, particularly or other animal species, especially with the hantavirus, which is very common in rodents, but potentially also West Nile or dengue, or even not virus histoplasma. So your thoughts about co-infections?
Yeah, so co-infections are definitely something that we've looked at. We've done a lot of rodent trapping. So as I said, we find Cocci a lot in the burrows. We've also done a lot of rodent trapping. We've been looking a lot at the microbiome. So there are definitely other onigonalian fungi that are present in the lungs of these animals. So co with Cocci. We don't know the implications of that at all. We haven't really looked at viruses much, but hantavirus is very prevalent here in Arizona. We have a lot of outbreaks here. So it's definitely something that's on our radar, but we haven't specifically looked at viruses yet.
Thanks so much, Bridget. Jay?
Okay, Frank, I'm actually going to take the chair's prerogative and ask the first question about the CGD work. So I'm not that familiar with the genetics, but I know, do you see differences in the inflammatory response or the epigenetic landscape if there's a misfolded protein mutation, missense mutation versus a premature stop where there's hardly any protein made in the cell?
Yeah, so we for that reason, we only did the FOX47 patients. We had 5 of them. And I know that I would expect that it's different because the GP91, they are more severe. They could have different effects. I think, although broadly we tested 1 and they also were very deficient in the functional aspect, but it wouldn't be surprised if there are differences between the mutations. But I think that the final common pathways would eventually, I don't think one goes into a completely different epigenetic landscape than the others. There might be differences in subtly, but not in the final big picture. So I think we would see differences, but not.
And then a quick question from Dr. Gaffin. Sarah is asking, do type 1 and type 3 interferons have similar effects, or is the effects somewhat unique to type 2?
Yeah, that's a very cool question, because I couldn't go into detail there, but there is definitely difference between the type 1, and knowing that the CGD patients are also lupus prone, so they are especially seeing type 1 interferon signatures there, much more like an interferonopathy. It's interesting. We are studying that at the moment to see how that works. They are different. So to answer that, they don't do the exact same function, Sarah, like the gamma interferon, but they do have effects in CGD that we are studying.
Great, thanks. Professor Gao.
So Bridget, I'm going to try and combine two questions which came in the chat. They're both about spores of Cocci, about the arthrospores and the endospores. So the first part of the question is, in terms of environmental transmission, your model implies that spherules or endospores can convert back to arthroconidia. And do you have any in vitro data for that transition? And what sort of signals are involved in that? And then the related question, which you might want to take at the same time, is which of these two infection routes, via the endospores or the arthrospores, do you think is the more common route to human infection? So the first.
First question, the evidence that we have for the transition from endospore or spherules back to mycelia and then arthroconidia, that's where we get most of our isolates. So we have spherules in the patient that form granulomas, and then those granulomas are, surgically excised or taken from necropsy. We take those, grind them up, and then they grow quite easily as mycelia on plates. The mechanism behind that has not been looked at all. We don't know anything about which genes are upregulated in those conditions. It's definitely something that we've thought about. The other end of the cycle is definitely more well studied, so that, arthroconidia to spherules transition, most of the investigations have looked at that transition because that's what's clinically relevant. There are some possibilities of infection with endospores, but those are all sort of contrived. So basically taking endospores, injecting them intraperitoneally into mice, and they will become infected, or intravenous introduction into mice. So we believe that, yes, the primary route for infection is that arthroconidia that are in the soil, in the dust, and you inhale those. Yep.
Thanks, Bridget. Jay.
Bernard Hube is asking about this viral-associated Aspergillus. Is the growth of Aspergillus affected by the tissue damage, and like do you see the same vascular invasion that you would see with a neutropenic host or bone marrow transplant, or is the growth of Aspergillus kind of different in the context of COVID?
Yeah, so that is a really important issue. We have not yet studied that, Bernard, and it would be great to do that. And I know you have the capacity to do these kind of things, so that would be great to do. And I can only speculate about what I would know, but it does matter what type of microenvironment is damaged there. That makes a lot of sense. If a cell loses the capacity to be doing what it should do, like pour out defenses, etc., it also pours out a lot of toxic stuff, and not to mention the neutrophils with the nets that have got a lot of attention there, which is a different and nice interaction. I would suspect it matters. Having said that, we have tried to see Aspergillus species with Palvaway in different influenza. So we are trying to study to see if it matters if you have different species and environment exposed to the Aspergillus, which might give you an answer if there is something different, because I don't think every microenvironment of a host is the same. So you would then expect that it grows differently. And second, I would expect that it matters where that happens. And you must probably have ideas about stuff that is in epithelial cells that might do that.
Well, I can tell you one thing difference between influenza and COVID, at least in the murine model, is that in influenza, your bronchoalveolar stem cells are intact and they actually proliferate. They have these keratin 5 regenerative epithelium. We don't see that in COVID, and we know that the bronchoalveolar stem cell actually expresses the ACE2, but I don't think there's too much sialic acid. So the tropism of the two viruses seem like they're pretty different.
They are, Jay, definitely there is a lot of difference in the setting of the endothelium being involved here. I haven't talked about the kallikrein kinin system. I know you did some work with Sarah before on the kallikrein and candida, etc. But we touched upon that also now in the setting of COVID. So we're trying some studies there. And it looks very interesting. I think the endothelial part or things very unexplored in this setting and now is reviving with COVID.
Thanks, Frank. Neil.
So Bridget, I'll try and bring in two questions from Elaine Bignell about the Pac-C work. And they're about the phenotypes of the mutants. So the first is, the Pac-C null mutant's got a yeast lock phenotype, so what can we expect of such a morphology in terms of pathogenicity? And she's pointing out in crypto, surprisingly, the altered cell surface of the mutant leads to heightened virulence. And the other phenotype, in another question, was that recent phenotyping in Aspergillus shows that the Pac-C is important for promoting Thermo Tolerance. So have you got any preliminary evidence of that type of phenotype in Cocci? So one's about pathogenicity and one about Thermo Tolerance.
Right. So those are really interesting and important questions. I was hoping that Elaine would be excited about the Pac-C work. And so for infection model, we're playing around with that right now, because again, I said that endospores aren't sort of a normal route of infection. So that's where we started doing maybe some IP route of infection or intravenous potential introduction of those yeast cells. And we are curious to see if that does actually enhance pathogenesis in the mouse model. So currently the strain that we are working with, that Silvera strain, is highly pathogenic. So if we infect with Silvera, the mice all die within 12 to 14 days. So we're actually starting to look at a lower virulent strain so that we can do some more fine scale measurements. So if we have something that's actually hypervirulent, we would be able to detect that more easily. So that's one avenue that we're exploring right now. And then the second question was about Thermo Tolerance. And it seems like it's not quite as happy at 37 degrees in the absence of Pac-C. But again, we need to thoroughly investigate that, but it is a very tantalizing observation. Yep.
Thanks, Bridget. Jay?
Frank, another question from Dr. Bignell about, what's the mechanistic basis of the interferon gamma action in terms of the metabolic status? So what is mechanistically, what is interferon gamma actually doing to affect the metabolic profile of the cells?
Yeah, that is obviously a question that we're looking into. And one of the things that we tried is to see how fast and does it require signaling or is it doing something else? So I can tell you, at least when you inject it immediately in a cell, it doesn't restore. It needs at least 24 hours. So there is a signaling pathway going on. Then second, we do know that that's probably step 1 dependent. So that's the question why it was so interesting to look at the type 1 interferons earlier in the question, but there are still differences which probably then have to do with the interaction of the stats. And it's a major hub, step 1, for changing epigenetics. That is one thing. The other thing is that we do know that all this glycolysis pathway, at least, is HIF1 alpha mTOR dependent. And we do know also that gamma interferon has a huge role there. And we are seeing already that that's one mechanism. And that flips back to autophagy, connecting a sort of a metabolic hub in a cell that all comes together. So I think these are so fundamentally processes that are all interconnected, and gamma interferon somehow does play a role at the redox, metabolic, epigenetics, and it's STAT1 dependent and signaling dependent.
Thanks. Yeah.
Thanks. Because of the time, I think I'll take another question and then return to Elaine's if we've got time, her third question. So this one's from Martin Waker, and it's about the distribution of Cocci. Bridget, so can Cocci species be found outside of the Americas? And is there any possibility that human activity has contributed in any way to the spread of the fungi around the world?
Yeah, that's a great question. So people have looked, not exhaustively, but people have looked for the organism outside of the Americas. There's no convincing evidence that we find it outside of North and South America. There is pretty interesting evidence that would suggest that potentially human migration from North America into South America may have introduced Cocci into South America, but that's still quite widely debated. So we've looked at some timing events. So when did the species or strains or populations arise in South America? And currently our dating it has a pretty wide margin of error. And so the exact mechanism for how Coccidioides emerged in South America is still unknown, but certainly there is a potential role for human migration.
Interesting. Thanks. Jay.
Frank, so in terms of immunotherapy, then you mentioned at the end several potential clinical trials, and like for a clinical trial to be successful, obviously patient selection up front is going to be really critical. So how would you envision selecting patients? What would the inclusion exclusion criteria be? Because obviously part of the problem, it's like sepsis, right? You don't know exactly where they are in the course of the illness, and so have you given some thought about what kind of groups of patients would you consider for the various interventions?
Yeah, so let me start where we talked about COVID and influenza. One of the ideas of the gamma interferon is also that at some point when you are deficient and you still have your innate immune system to do the job, but your T cells are gone and there is not a good backup anymore, that reflections in the clinics could be, one, we could measure the HLA-DR expression on the monocytes, but second, we can look if there is herpes reactivation, which is one in the clinics when we do a BAL, very clear to me that there is a dysfunctional T cell compartment starting. Then third, if there is a sign of the Aspergillus, if you do the definition of kappa, all those together, probably you then start selecting patients who are in the immune status of gamma interferon deficient, don't have the T cells to do it. And I don't think then you are only treating kappa, but I think you're treating the host status. So then the virus, the herpes virus reactivation, the HLA-DR, they all should come back. So we have now defined a patient that we would treat with gamma interferon 1, persistent viral load for COVID, HLA-DR expression low, virus herpes reactivation, and definition of kappa. That is a status that I would start with in COVID.
Great. Thanks for that, Frank. Neil.
Yeah, we're getting close to the half hour, but potentially quite a quick question from Duncan Wilson to Bridget, which is, do you know of any examples where Coccidioides has been found in the same sample that you took along with histoplasma?
So from soil samples, I'm assuming, or
yeah,
from,
I think it's any sample.
Any samples.
Probably environmentally having the two organisms in the same
Yeah, so there's not a lot of overlap, which is interesting, and I think that's probably what Duncan is curious about, is that pH limitation. Yeah, so I think it's definitely playing a role. We almost never see overlap in the environment, and we so far have not seen overlap in any of the host lung tissue from the rodents that we've been collecting. Yeah.
Okay, thank you. I think that may have to be our last question, Jay.
Yeah, I'd like to thank both speakers for excellent talks. Really enjoy them.
Nice.
Oh, thank you.
Okay, see you everybody.
We look forward to see you all at the next MycoTalks.
Yeah, like.
Bye-bye.
Thanks again, everybody, and thanks to our speakers.